Genomic analysis conducted with advanced algorithms presents a potential for a number of rapid advancements in the way we understand the body and construct our medicines. Where we were once left to diagnose issues based on outcomes, combining genetic testing and blood sampling can allow predictive modeling to identify potential pathways of disease well before they start. Using methods as simple as comparing available nutrients to numbers of copies of particular genes, we can identify potential nutritional shortfalls that can be shored up well in advance of issues arising. Inserting AI analytics into the diagnostic and particularly genetic pathways also allow for hitherto unforeseen specificity in diagnosing and treating the root causes of issues that have plagued humanity for eons, by understanding how genetic expression can be altered via the introduction of environmental contaminants. CRISPR with deeply paired AI opens the door for massive specificity in gene splicing, where massive blocks of DNA are used to select for single base pair edit locations when matched with rapid shotgun sequencing. Further combining comparative analytics across large databases of genomes, we can shrink the storage space needed for large genetic datasets by statistically tracking areas of the genome with large degrees of mutability, and matching individuals based on their mutations in areas within differential levels of conservation across a species and subgroups. These and many more pattern recognition paradigms available now open the doors to not only diagnosing and curing disease, but as well using the DNA directly to store information and perform data operations on large data sets using inbuilt cellular mechanisms to do what would have been unthinkable just a few years prior. We can as such create new services to help extend lifespan by means of monitoring telomeres during regular checkups and tracking differences in genetic aging and mutation across populations. This leads to advanced warning of new emerging environmental carcinogens while offering direct points to reverse environmental genetic damage through the use of historical genetic datasets. Searching sequences to create better cutting selections opens massive new potentials in crafting new reversible genetic and epigenetic therapies, and in all likelihood preparing humans for long-duration space travel by splicing in traits that would help us endure long-term hibernation and increased radiation exposure. These enhance feature translations by splicing into genes to add or remove coding pairs and sequences using extra-long specific cutting codes opens the door to using genetic storage systems, and also creates the potential for fractal memory redundancy using DNA to store information that encodes massive amounts in live cell cultures or soluble crystals.